this brief video, I'm going to demonstrate how you can use an illustration like this to pretend that you are an expert in this area and find biomarkers that you want to study in your virtual experiment for my biostatistics class. So here we see we're at the website QIAGEN. They have this lovely genes and pathways, these beautiful ornate illustrations. You can register and download PowerPoints and the images and things like that. But more importantly, what I want you to do is to take some time to find one. I should, in a moment, I'll try to get you to the page that has all of them. It used to be a little bit easier in my mind anyway. But you can read about it here. So it gives a great deal of information about the process. I currently am working on angiogenesis, but focus on some of these markers here and how endothelial cells stick to their basement membrane, or maybe to each other, or whatever it might be. Through my searching online, this model system came up, so I thought it would be a great opportunity to do this. I'm interested in this part here, and maybe 3 or 4 of these. So I'm gonna try to pick and choose the things that I'm going to track in this pathway, and then we'll find data on them through pulling down gene annotation tables and transcriptomics from NCBI GEO to generate some virtual data and perform statistics on it, just as if we had run some analyses to track the messenger RNA for these in the cells. And of course, you'll pick your study on the NCBI GEO. Anyway, when you click on these, it gives you the gene names. So this is what I want you to get, is the gene symbol. So you can select copy and paste. What I'm going to do now is take you to another. You can start a list in Word or something like that. And now I'm going to go to NCBI GEO. You can't see that, but anyway, just typing it in. And here you see it. I just did a Google search for NCBI GEO, and then you come to this. And that isn't what I wanted to do, sorry. That's how you get to NCBI GEO. What I want to do is do a search for my gene. I'm doing Control V to paste in my gene. Let me do this. Look, there's all my pictures. That's how I found this in the first place. And then space, and I type in gene card. You see it right here. I did the search. So here you see the ITGA6 gene at gene cards. There's other sources, but I like the gene card because this gives us the human gene. And then this gives you information about the gene and the function of it. And summarize, that gives you a tremendous amount of information. You definitely should become familiar with this. It just goes on and on and on and on. Quite amazing. Okay. So it's a good resource, but I come here to make sure that this is the symbol that's currently being used for the human genome. And we make our list. You can supplement it however you want. If you've heard it called something else in the literature, its alias might be in here. If you do a search, the alias might be here, and then the new, most current gene symbol will be there. And then here's the integrin alpha 6. So a couple lessons from this. We see a single name, the integrin alpha 6, and then it's with a beta 1, but just the showing one thing here, and you get 2 different genes for it. So why, right? What does it mean? And you can see here it shows the transcripts. These indicate the location on a DNA microarray. anyway, different ways of referring to the genes and things like that. Some orthologs here. You can get gene lists from this site as well. Right now I'm just trying to have you use the gene card as well to get some information from there. Just quickly, while we're here at the NCBI GEO, you can search if you're interested in diabetes. You can say I've searched diabetes and Alzheimer's, wondering what's going on there. So you look for a study that might be interesting to you, or more so that would have, well, anyway, be of interest to you for what we want to do. So here's one in the rat. This gives you some information about it. You can see the gene annotation that they used. It's more of the platform for the microarray that they used. You can click on that and get information about that. And then here's the series reference for the samples. This gives you information about that. Now in each of these, what I recommend that you do is you do a file print. I'm in browser, blocking on the name right now, speaking of Alzheimer's disease. Anyway, you can see that you can change the printer to the save as a PDF, and then you can save it in a folder and start getting these on thumb drives or some folder that you have somewhere. Before you do that, you want to come here and expand so it'll list all of your samples. And what you'll do for the class is get the text file. You'll just click on the series matrix and click here, and of course it'll just download it. And we'll have to see hopefully we can unzip it in class. If not, we can just get some numbers to work with by clicking on a sample. It shows the table, and we can view the full table. And once the full table is here, might take some time. They're kind of big. We begin selecting. Hold down the shift button. Scroll over here. Click to the bottom, and Control C or right click. And gotta give it some time to, there you go, copy it and then paste it into Word. And just paste it in. It'll take a little bit. These are kind of big. It's going to take us some time there. So that they're big files, and that's what we're after, of course. There it is. So, excuse me, once you have it in Word, and you just do a file, save as, plain text, right? That's the tab delimited in Word. Then you call that into Excel, and it'll be, if you paste it into Excel, it may not paste just right, but I put it into Word and save it as a text, and then I open it in Excel, and then Excel parses it based upon the tab delimited separations. Long way to the top there. And then we can work with it there. It gives us one. We can do that with 2 or 3 of these if we can't unzip the whole file. Anyway, that gets us a database, a data set for gene expression profiles. And then you will have to go to the GPL. This is the platform. because we'll take it into a database. And when you come down here, usually they have a download the full table. And when you download this full table, it just comes as a text file, so we can open this right into Excel, no problem. But this .gz, the zipped one, we may have to expand it. So now let's come back and see if we can get to the whole listing of the pathways. So search for your pathways, whatever species you want. If you don't know, just go to Pathway Central. Cell cycle regulation. Okay, so what do you want to look at? It's all here. Maybe not everything in the world, but a lot, right? I'm still going. Look at them right over here. I've seen a couple that I might want to take a look at. Okay, so study any one or more pathways. You just need a gene list. That's the task for today. And find the database. Find the data set, the transcriptomic data set on the GEO with your search. And let's see if we can get the whole data set pulled out. If not, we'll just pull down separate ones and create at least 3 samples. I guess we'd have to get 6, 2 groups, 3 in each. But anyway, we'll see what we can do in class about unzipping these. Okay.